Oogenesis, pronounced like that, I think. Oogenesis is like spermatogenesis, except we're talking about the formation of egg cells, or ova, O-V-A, which is plural. Singular would be ovum, O-V-U-M. Oogenesis is similar to spermatogenesis in terms of the meiosis process, but it's a little bit more tricky because there are a few stop and pause points. That's about it. So let's take a look at the general structure of an ovary, and in this case we're looking at a rabbit ovary because this is one that's often used as an example of ovarian tissue. So you should know that oogenesis is the production of a ovum, and obviously it occurs in the ovaries, whereas sperm production occurs in the testes. So if here you see an electron micrograph image of what an ovary looks like, here's a diagrammatic form. So obviously you need a blood supply to be able to transfer nutrients and oxygen to keep these cells alive and dividing. You have an outer layer of germinal epithelium cells. Epithelium just refers to the outermost layer, germinal meaning having to do with producing new cells. The cortex here, this area, the cortex, now in all kinds of body tissues you'll see these words medulla and cortex being used. You see this in the kidney as well too. So the cortex, if you see the word cortex, it doesn't mean it's specific to the ovary or specific to something else, specific to anything else particular. But if you look at all this stuff, you should be able to figure out which particular general area we are referring to. And then a secondary oocyte inside here is when it goes from the primary oocyte, you'll see this later, primary oocyte to the secondary oocyte, then you'll start to be able to see a follicle, a mature follicle with the ova being ready to be released, depending on what stage we are at. So another general diagram. So going around in the beginning, you have an unending source of cells which are diploid, meaning they have 2 times n equals 23, so 46 chromosomes. They continually divide by mitosis to produce more and more diploid cells that are all 46 chromosomes. These diploid cells will get larger, and when they become larger, we call them primary oocytes, but they have not yet undergone any kind of division, so they're still diploid, or 2n at this point. The primary oocytes start the first division in meiosis, but they stop in prophase 1, and little baby girls, when they are born, they have already a whole bunch of these all ready to go, but they've all stopped in prophase 1, and they won't continue through the next stages of meiosis until they hit puberty and the menstrual cycle actually starts. So they're roughly born, we say, approximately 200,000 in each ovary, so 400,000 primary follicles ready to go. Of course, it's impossible. There's no need for 400,000 ova. Well, maybe there is, depending on what you look at. I mean, per ejaculation, men are releasing 500 million sperm cells. So is there really a need for that? But in terms of genetic diversity and the chance that any one of them is going to succeed is very low, so we do have this happening. But per menstrual cycle, only one of these usually gets released. Okay, every menstrual cycle, a few of those primary follicles develop, and the primary oocyte completes its first division of meiosis. So now we actually go from diploid to full haploid. So now we're talking about 23 chromosomes, and we're already down to unique cells, unique ova. The cytoplasm of the primary oocyte is divided unequally. That's important because at the end of all of this meiosis division, you're only going to end up with one big egg cell as opposed to 4 egg cells. When this happens with the production of sperm, you end up with 4 sperm cells that are really tiny. But at the end of this whole process in oogenesis, you end up with one larger ovum. And the others, what happens to the other side that divides? Well, it kind of degrades and turns into this thing called the apolar cell, and you'll get another one later after the second division, or 2, depending on if the polar cell continues to divide or not. The secondary oocyte starts the second division of meiosis, but here's another pause. The other pause happens in prophase 2. So the first pause happens in prophase 1, the second pause happens in prophase 2. Now, when the follicle actually bursts, then the ovum gets released. Remember, this will be timed closely with the peak in LH. LH is luteinizing hormone, which reaches a peak approximately 2 weeks after the menstrual cycle begins. So when LH peaks, then that's when the egg is going to be released. So that's when ovulation usually happens there. And the egg will not complete the second division of meiosis until it has met the sperm cell for fertilization. After fertilization has actually occurred, then the second division actually completes, and then you end up forming the second polar body, and then they are of no use anymore, so they degenerate and break down. Here's a summary of oogenesis. It's a similar diagram to what we saw in spermatogenesis, except that we show the polar bodies in the end. So here we go, mitosis. These cells are just continually reproducing themselves. They're still diploid. Turns into the primary oocyte just after growing larger, after first division of meiosis. Okay, remember there's a pause here, prophase 1, and then during the menstrual cycle is when this actually starts again. And then the uneven division of cytoplasm produces one larger cell, haploid cell, and a small haploid polar body that disappears. And after the second stage of division, right here, you end up with an additional cell. But in the end, here you end up with one large ovum there. Okay, one large ovum. So this divides and then turns into this. This is getting bigger and bigger at each stage, and the polar body is going to degrade right there. A good thing to do is to compare, draw a diagram, do a Venn diagram, compare spermatogenesis and oogenesis. I mean, they both involve meiosis. They both involve the formation of primary spermatocytes and primary oocytes, but there are a few specific things. Sertoli cells and spermatogenesis, you should be able to list at least 4 or 5 things that are different about them. But of course, when you see this word compare, although even if you're not asked to compare and contrast, if you just see compare, they are asking you to outline differences as well as similarities. Okay, so make sure you catch all of that. All right, post any questions that you have.